Hi everyone. So today I'm going to be going over the mitosis lab. Um, I didn't have enough resources to film an actual virtual demo, so I'm just going to be going over the general background knowledge, the procedures that we did in class, and then I'm going to share some results with you guys so you guys are able to complete your lab log. All right. So I'm going to begin by reading over this little paragraph here. We're going to be talking about mitosis. All right. The process of growth and division in a typical eukaryotic cell is called the cell cycle and is composed of two phases, interphase and the mitotic phase. Each cell cycle begins with the formation of a new cell and continues until that cell divides into two offspring cells. Each offspring cell then begins the cycle again. Although the phases of the cell cycle are fixed, the amount of time spent in each phase, especially interphase, varies among different organisms and among different cells within an organism. In the apical meristem region of an onion root tip, one complete cell cycle is typically completed in approximately 24 hours. Of that time, between 2 to 4 hours is spent in the mitotic phase of nuclear and cellular division. All right, so from that, we can obviously make the connection that for onion cells, the majority of time is actually spent in that interphase phase. All right. So interphase can be subdivided into three phases, G1, S, and G2. During interphase, normal metabolic activities such as the synthesis of RNA, proteins, or other cellular macromolecules, and the building of organelles cause the cell to grow in size as the cell prepares for its next division. In the apical meristem region of an onion, interphase lasts about 90% of the total cell cycle, so around 20 to 22 hours. During the G1 phase, GAP1, the cell is metabolically very active as it grows following the previous cell division. In an onion root tip, cells outside the apical meristem go into an extended G1 phase and rarely ever divide again. This is called a G0 phase. All right. So the G0 phase is basically just a cell doing its normal thing without preparing to divide. Okay. Moving on, from the G1 phase, meristem cells enter the S synthesis phase of interphase. In the S phase, an exact copy of DNA is made in the nucleus of the cell. Additional proteins, RNA, macromolecules, and organelles continue to be made during the S phase and also in the final phase of interphase, which is called the G2 phase, GAP2. The S phase is the longest phase of interphase, usually taking between 10 to 12 hours to complete. Okay. The dramatic events of nuclear division take place as part of the mitotic phase or M phase. Chromosomes coil and become visible with a compound microscope during the mitotic phase. The M phase is subdivided into two parts, mitosis or nuclear division and cytokinesis, which is cell division. Cytokinesis occurs simultaneously at the end phase of mitosis. After cytokinesis, each offspring cell enters G1 and the cycle begins anew. Okay, so what I want to highlight here is the M phase of your cell cycle includes mitosis and cytokinesis. Cytokinesis is not a part of mitosis. It happens right after, but it's not included in mitosis. Your four main stages of mitosis are prophase, metaphase, anaphase, and telophase. Okay, so mitosis is subdivided into five phases. During prophase, the nucleolus fades and chromatin condenses into chromosomes, and cytoskeleton microtubules create the spindle fibers necessary for the chromosome separation. In prometaphase, the nuclear envelope breaks down, so there is no longer a recognizable nucleus, and spindle fibers elongate. During metaphase, the chromosomes reach a position called the metaphase plate, which is midway between the poles. At the onset of anaphase, the sister chromatids also separate, splitting the chromosome in half. The spindle fibers shorten and drag the attached half chromosomes or chromatids to opposite poles of the cell. In telophase, the chromatids arrive at the poles and the spindle fibers that have pulled them apart disappear. A nuclear envelope reforms around each cluster of chromosomes, and these chromosomes return to their more extended form while cytokinesis begins. Okay. So what would happen if the rate of mitosis was altered in an organism? You guys should learn about this in lecture, but one of the main things that can happen is uncontrolled cell replication, which is known as cancer. Okay. What are some factors that could affect the rate of mitosis? There's a few things here, but one of the big factors that you guys should have talked about in lecture is hormones, and that's what we're going to be playing around with today. Okay. So this part right here is going to go into what hormones do and how they can affect onion mitosis. All right. So I'm not going to read all of this out loud, but basically what happens here is we're going to talk about a hormone called gibberellic acid. Gibberellic acid, basically what it does, it ups the rate of cell division that occurs in the meristem region of an onion root tip. Okay. So what we did for this experiment was we took onion roots, which were grown in gibberellic acid, which is again this hormone that can basically up the amount of cell division happening in onion roots, and then we grew onions in regular water. All right. Then what we did is with these tips, we stained them, looked at them under the microscope, and actually saw if the amount of cell division happening was different. Okay. So I'm gonna show you guys first some pictures of what the roots looked like under the microscope, and then I'm going to show you guys actual counts that my students did last semester. Okay, so before we do that, there should be a few things you want to look at first. So remember, what we're looking at here is a hormone. Essentially, what we should see is the onion root that was grown in this gibberellic acid should have more cells in mitosis, so actually dividing, than an interphase. Interphase is just preparing to divide. So if you put a hormone into an onion root tip that ups the rate of mitosis, you should see more cells in their actual mitotic phase. So you should see cells in prophase, metaphase, anaphase, or telophase. Okay. And then when you look at the actual onion root tip that was just grown in water under the microscope, you should see the majority of those cells in interphase. So you won't be able to differentiate what steps of mitosis it's in. Okay, so I'm going to go share my screen and show you guys a few pictures. Okay, so the pictures that I took in lab on Friday were just a little bit too blurry for you guys to actually see the different steps of mitosis. So I have two pictures here that I'm going to show you guys. This is what mitosis looks like in onion root tips. So interphase, premetaphase, metaphase, anaphase, and telophase. All right, so this is what they look like under the microscope. What my students did was when they were counting the amount of cells that were in that mitotic phase, they included any cell that looked like it was in any of these stages. Okay, so they weren't looking for a specific stage in mitosis. They were just looking to see if that cell was actually in the mitotic phase. Okay. So like I said, the ones grown in gibberellic acid, the majority of them should be in this one of these stages. All right. And that's just because, again, gibberellic acid ups the rate of mitosis. Okay. So this is another picture. This is more closely related to what they actually look like in lab since we dyed them using a blue dye. Anything after this first picture is in mitosis. Okay. So when we're looking for interphase, my students were looking for this dark ball that wasn't really able to differentiate any specific chromosome. This is an interphase. Okay. So what we should see is the onion roots that were just grown in regular water. The majority of the cells would be in this phase right here in interphase because again, it's a normal mitotic rate. Nothing was put in to kind of up the rate of mitosis. So the majority of the cells are still in interphase because again, interphase is that longest stage of the cell cycle. Okay. So now I'm going to move on, show you guys actual data that my students collected. Okay. So this is from a few semesters ago, if I'm not mistaken. And as you can see here, are the counts that they did. Okay. So this is the control. This is again, the onion roots that were grown in water. And as you can see, the number of cells in interphase are a lot higher than those in mitosis. And again, that makes sense because again, water shouldn't up your mitotic rate. So the majority of your cells should be in interphase. All right. Then on the right here, this is gibberellic acid, just as a chemical formula. And as you can see, the amount of cells in interphase here are a lot lower than the ones in mitosis. Okay. So what you're going to do with this data is you're going to use these numbers down here to create a graph to represent the difference in the cells in interphase versus mitosis for both treatments. Okay. So you are comparing, in this case, the control, so the water, to the gibberellic acid. So this should be a bar graph. Okay. With that, you guys should be okay to complete the experiment. As always, you can consult with Camila if you have any questions. Yeah,